Bismillahirrahmanirrahim. Assalamualaikum and a very good day I bid to everyone. So my name is Muhammad Ukasha Irfan bin Marahimi. My metric card number is AW180179.
Hi, I'm Nur Iska Ashiki binti Muhammad Najib.
So today, me and my friend gonna tell you about Laser Raman Spectroscopy for sample analysis.
Raman spectroscopy is a popular technique for the analysis of molecular structure and is considered complementary to infrared spectroscopy. Raman spectroscopy is based on the Raman effect, which was first identified by the Indian physicist Chandrasekhara Venkata Raman in 1928. The Raman effect is based on scattering of light, which includes both elastic and inelastic scattering. When a sample is exposed to a monochromatic light in visible region, the sample absorbs light and major portion of the light get transmitted through the sample. However, a minute part of the light is scattered by the sample in all the directions. One can observe the scattering at right angle to the incident beam. If the scattered light has the frequency same as the incident light, then scattering is called Rayleigh scattering. However, it has been observed that 1% of the total scattered intensity occurs at frequencies different from the incident frequency. This is called Raman scattering. Raman scattering can be thought as a two-photon process. The electrons have different vibrational levels. They are defined by specific energy differences. When an incident monochromatic light interacts with an electron in the sample, the electron absorbs energy from the incident photon and rises to a virtual state of energy. Raman scattering is about 1,000,000 times less intense than Rayleigh scattering. Therefore, to obtain Raman spectra, it is necessary to prevent Rayleigh scattering from overpowering the weaker Raman scattering. Raman spectra are measured by exciting a sample using high-intensity laser beam, with the resulting scattered light being passed through a spectrometer. The Raman shift is the energy difference between the incident light and the scattered light. In the resulting spectrum, the vertical axis is the intensity of scattered light and the horizontal axis is the wave number of the Raman shift. The Raman shift is associated with two different energy bands. The shift at wavelengths higher than that of the incident light is termed as Stokes scattering. The shift at the wavelengths lower than that of the incident light is termed as anti-Stokes scattering. If the frequency of scattered photon is less than frequency of the incident photon, Stokes lines are observed on Raman spectrum. This happens when the electrons absorb energy. Similarly, when frequency of emitted photon is greater than incident photon, anti-Stokes lines are observed. This means that energy is released by the electrons. The Raman spectra gives the molecular fingerprints, and it is different for different molecules.
So generally, there are three primary components for any Raman spectrometer, which is the excitation source, sampling apparatus and a detector. So moving on to the Laser Raman sample preparation. But before that, we may be wondered what type of sample that can be characterized using Laser Raman. So it can be either solid, liquid, gel, gas, slurry or even powder. So as we know, Laser Raman spectroscopy can easily and effectively operate on a flat surface of the sample. So before we go any further for the characterization part, we need to polish the sample in order to obtain a flat surface. The fact that the sample surface cannot be covered or cannot be coated by any other substance, for example like gold or carbon coating as done on the SEM or EPMA. So if the sample surface are coated, so we need to remove the coated layer by simply using the mechanical rubbing with the organic solvent to polish the sample to remove the coated part. So ladies and gents, when we talk about Raman spectrometer, of course, this Raman spectrometer will use the laser as their source. So the laser choosing need to be considered before any characterization taking part. So as you can see in the screen, this is the typical example of laser that has been used in Laser Raman characterization. For the first one is ultraviolet, with the wavelength between 244 nanometer until 364 nanometer. And then we have visible, which is the wavelength between 457 nanometer until 660 nanometer. And last, we have near infrared with the wavelength between 785 nanometer until 1064 nanometer. So the correct choice of laser wavelength will determine the effectiveness of the laser Raman, because we know that different wavelength will produce different energy. Also here, I'm going to tell you the difference of the different laser wavelength that has been discussed just now. For example, blue or green laser from visible light can be good for inorganic material and resonant Raman experiment. For example, for carbon nanotube and other carbon material, and even in the surface enhanced Raman scattering (SERS). And then we have red or near infrared. Basically, these lasers are good for fluorescence suppressions. And also we have the UV laser, ultraviolet laser, for resonance Raman on biomolecule such as protein, DNA and RNA. And now, moving on to the experimental part. So as you can see in the screen, the figure shows you the schematic diagram for Raman spectrometer. So the display setup focuses the illuminating laser, the one in the green color, down to a line on the sample, the slit scanning mode, which also can be replaced by a spot by removing the cylindrical lens. Also, in the slit scanning mode, many spectra are acquired simultaneously. So each position along the line on the sample will produce a spectrum along the CCD detector. Now, we will talk about the application of laser Raman in industry. So usually, laser Raman are used in geological characterization of mineral phases that cannot easily be identified under the microscope. So there are a few of minerals that can be successfully identified using the Laser Raman spectrometer or spectroscopy. For example, the mineral in bauxite and laterite, or iron ore minerals, carbonate phases, manganese minerals, and there are many more that can be used to characterize using the Laser Raman. Next, laser Raman also can be used to identify the vein forming fluid through the fluid inclusion study. So basically, fluid inclusion are microscopic entrapment of liquid and gas within the crystal. So the identification of inclusions will be in the transparent germs in variety of minerals. So why laser Raman is used in this characterization? This is because the laser beam can penetrate in a transparent mineral without causing any harm to its structure or composition. So this laser is used to penetrate the germ variety of minerals or crystal and collect the spectrum from the mineral inclusions inside the gemstone. So laser Raman also used in shock mineral study or characterization, which this study will depend on the molecular vibration which is affected by the lattice deformation. So the laser Raman spectrometer has been successfully used for study of shock metamorphic polymorph in meteorites and impact craters.
Conclusion, Raman spectroscopy is useful technique for identification of wide range of substances such as liquids, solids and gases. It is a straightforward non-destructive technique requiring no sample preparation. Raman spectroscopy involves illuminating sample with monochromatic light and using a spectrometer to examine light scattered by the sample. In modern Raman spectrometer, lasers are used as a photon source due to their highly monochromatic nature and high beam flux. Lasers used in Raman spectroscopy range from the UV into the near-infrared and beyond. Different wavelengths region offer advantages and disadvantages, with the final laser selection largely dictated by the sample being studied. Some samples can be analyzed at any wavelengths with no issues, for example toluene, but many samples' laser wavelength choice is crucial for high-quality Raman spectra, for example given its polymers with high fluorescence backgrounds.